Hi everyone, my name is Valeria. So my title is very general, but actually I'm going to be talking about unsupervised approaches and how they can be useful in the field of psychiatric neuroimaging in particular. So first I'm going to start with explaining the difference between supervised and unsupervised approaches and how they can actually be used to answer different questions. Then I'm going to go into one approach in particular called partial least squares, and I'm going to start with explaining the goal of this of PLS. Then I'm going to explain to you how to compute it step by step, and then how to interpret the results. And I'm going to illustrate this with an example with a psychiatric neuroimaging dataset. And then finally, I'm going to also give you an example of how to combine PLS with a supervised approach such as classification. Oops. Okay, so great. Okay, so a classical example in machine learning is you have pictures of cats and dogs, and each of these pictures is labeled as being either a cat or a dog. And what you want to find is a function that enables you to predict the label of your data. In unsupervised learning, you have a bunch of data which is not labeled, and what you want is to find some kind of hidden structure inside your data. And so one example of this would be to use a clustering approach, which would, for example, separate animals that have floppy ears from those with pointy ears. Or another example could be of unsupervised approach would be to put all the animals on a spectrum from brown to gray color. So as you can see from these two examples, these unsupervised approaches are more exploratory than the supervised ones. So when is actually unsupervised learning useful? So for instance, when your classes or your labels are unreliable or not well defined. For example, we know that in psychiatry, the way we categorize disorders today may not be the best or the most relevant to reveal the underlying characteristics in these disorders, as shown by the large overlap in the clinical symptoms or the genetic risk factors. And so in this case, in this field, unsupervised learning may enable you to find some underlying characteristics in your data, if it could show it, yeah. Such as, and maybe find some new categories or new dimensions which could allow you to, which could cross the boundaries in psychiatric disorders that we have today, and that could be more relevant to characterize these different disorders. So now I'm going to go into partial least squares. So PLS, in just a sentence, it finds optimal associations between two matrices X and Y. And there are different variants of PLS in the literature, some of which I've put in this table. And the main difference between these approaches is the kind of data that you want to put into your X and your Y matrix, whether it's imaging data or behavioral data or group labels or contrast, etc. But in this talk, I'm going to be talking about behavior PLS in depth. And behavior PLS tries to associate imaging measures with behavior measures. Sorry? All right, so imagine you have a set of subjects, and for each subject you have some imaging data, for example, functional connectivity data, and you have behavioral data. And those are two views of the data, and PLS will try to find the shared information between those two views. And so in this case, it will try to find patterns in your imaging data which are related to patterns in your behavioral data. And it does so by computing low-dimensional latent variables which show maximal covariance. So if you look at these two matrices here, you can see that they're in lower dimensions than the original matrices, and they show maximal covariance between each other. So your two views will be associated via different, the different associations between your two views are going to be called components or modes. And this could work. And each of these components is going to have a weight for each of your imaging variable and a weight for each of your behavior variable. Oops. All right. So imagine you have two matrices, X and Y. In your matrix X, you have your imaging data with subjects in the rows and connections in each column, and your behavior data in the Y matrix organized as subjects by behaviors. So what PLS tries to do is it will find weighted pairs of vectors. Yeah, it's not working very well. Okay. So PLS is going to find weighted pairs of vectors, and these matrices, these weighted pairs of vectors will be contained in matrices V and U, which are called saliences or loadings in principal components analysis. So in matrix V, for instance, you will have a weight for each connection and for each component. In matrix U, a weight for each behavior and for each component. And then by projecting your original imaging data and your original behavioral data on these salience matrices, you get your latent variables, or also called scores. And for each subject, you're going to have a different score for each component on the imaging side and also on the behavioral side. So the way you compute it step by step, so you have your X matrix containing your imaging data as before, Y matrix containing your behavioral data. First, you start by normalizing the data by z-scoring both matrices. Then the first step is to compute a cross-covariance matrix called R, which will contain the strength of the covariance between all the behaviors and all the connections. And then the main step of PLS is the singular value decomposition of this cross-covariance matrix into low-dimensional matrices U, S, and V. So we've seen U and V before. Those are the salience matrices containing the weights for each behavior and for each connection by component. And then the matrix X, S is a diagonal matrix containing all the singular values. And we're going to go back to the singular values in just a minute. And then the final step of PLS is to compute the latent variables by simply multiplying your original data X on your saliences and your original data Y on your behavior saliences. So because PLS relies on singular value decomposition, the number of components is going to be the rank of the cross-covariance matrix, which is the lowest of the dimensions between imaging and behavior, which is usually behavior. The second thing is that the components are going to be ordered by the effect size, or the amount of covariance each of these components explains. So the way to compute it is simply to take the singular value in this S diagonal matrix, to take this, the squared value of the singular value, and to divide it by the sum of all singular values squared. And then finally, all the components are going to be orthogonal, meaning that each component will explain a different part of the covariance between imaging and behavior. Now for the interpretation, so you computed your PLS, the first question you're going to ask yourself is, are the components that are found significant? And to know this, you'll do permutation testing over the singular values. Then you want to know, for example, if you have different subject groups in your data set, you want to know whether the components that you got are differently expressed by groups. And for this, you can simply compute t-test on the latent variables between different groups. And then the main question, I guess, you're going to ask is how to interpret these components. What is driving each of these components, and which imaging and behavioral variables are driving these components. And for this, you compute correlations between your original variables and your latent variables. And finally, you want to know which saliences or which weights in your behavioral data or your imaging data is stable. And for this, you'll proceed with, we do bootstrap estimation of the saliency standard error. So for the significance of the components, as I said before, you do permutation testing. So usually what you do is you permute the rows of your behavioral data matrix, you compute the cross-covariance matrix, which you decompose, and then you keep the singular values in each iteration in order to build a distribution of the singular values under the null hypothesis. And in the end, for each component, you will have a distribution of the singular values that you got from each permutation of your data, and you can compute a p-value by simply by summing all the singular values that you got from a permutation that were higher than your original singular value, and you divide it by the number of permutations, and you just add a 1 on both sides, and you have a p-value for each of your components. Then you want to know if your components are differently expressed by groups. So here, for example, you have one component with subjects from 5 different groups. You have the brain scores and the behavior scores. So these are their latent variables. And so you can compute differences between the means of these brain and behavioral scores between groups, and you can find differences and see that, for instance, here, the healthy controls here show significantly lower brain and behavior scores than all of the groups, all of the patient groups. Then to know the contribution of the original variables to each component, as I said before, you compute the correlation between your original variables and your latent variables. And these are called structure coefficients in regression analysis. So for both imaging and for behavior, you will have a different correlation for each of your connection and for each of your behavior per component. And finally, to assess the stability of your saliences or of your weights, you do a bootstrap estimation of your standard errors. So what you do is you sample with replacement the rows in both your imaging and your behavioral matrices a certain number of times. You recompute the cross-covariance matrix, which you decompose, and this time you will you'll keep the values in the U and the V matrix, which are the saliences, in order to be able to compute the mean and the standard deviation across all your bootstrap iterations. And in the end, what you do is you compute a bootstrap ratio, which is the salience of each element of each connection and of each behavior divided by the standard deviation of this salience. And you get some sort of z-score. And this way, the connections and the behaviors that have a high weight or high salience and that also have a low standard error are considered stable. So now I'm going to illustrate this with an example of some data that I'm going to be presenting on a poster next week. So hopefully all these different steps will be a little more clear. So I'm using data from the UCLA Consortium for Neuropsychiatric Phenomics, which contains both healthy control subjects, but also different psychiatric populations. And on the imaging side, we have functional connectivity data coming from correlations between the time series of 400 cortical ROIs, to which we added 19 subcortical ROIs. And this gives us around 80,000 connections for each subject. Then on the behavioral side, we have 54 behaviors which contain clinical symptoms, but also cognitive measures and personality measures. So we compute PLS with the steps that I've described before, and we do permutation testing, and we find 3 significant components. And I'm going to go into 2 of them today. So I'm going to start with component 2, simply because the correlation on the behavioral side, all the top measures are positive, so it's going to be a little easier for the interpretation. So as I said before, we have 54 behavioral measures. Here I'm showing you the top 15 ones. And if you look at the measures that we have here, we can see that they're spanning various different cognitive domains, such as language, memory, or executive functions. So, and they all have positive values, meaning that if you load higher than on this component, then you have better cognitive performance. And this better cognitive performance is correlated to patterns in your imaging data. So the patterns here on this side, the positive patterns, for example, between the somatomotor network and the default mode network, executive control network, are positively correlated to better cognitive performance. And the patterns in blue are negatively correlated to this better cognitive performance. And then if you look at the saliences that are stable in this component, and if you compare it to the matrix that I've shown you just before, you can see that among these connections that are that contribute the most to this component, a lot of these connections are also stable across different resampling of the data. Now, if we go to another component, component 1, you can see on the behavioral side that it separates measures that have both a positive correlation from a negative correlation. If you look at the measures that have positive correlations, there are different measures of psychopathology, such as impulsiveness, anxiety, depression, ADHD symptoms. And in the negative correlation measures, we have control and 2 measures of memory, meaning that if you load high on this component, then you will have more symptoms, and you will have also lower control and lower memory performance. And this is correlated to these patterns here. So if you have more symptoms, then you will have higher connectivity between your dorsal attentional network and somatomotor network with the subcortical regions, and a lower connectivity within your somatomotor network. And same here for the stable saliences. You see a lot of the connections that are important for this component are also have a stable weight. And so finally, I'm going to show you briefly show you how you can combine PLS with a supervised learning. So you can use PLS to extract features that hopefully are biologically meaningful, and at the same time, it also allows you to reduce the dimensionality of your data. So for example, if we go back to this first component that I've just shown you before, which is related to psychopathology, you can use the scores of this component to predict the prognosis of these patients. So for instance, if we take just 2 features, the brain and the behavior scores of all subjects, and then we use these 2 features to classify patients with a better prognosis versus a worse prognosis, then we end up with 2 features that are at the same time both very low dimensional and that have biological relevance because they're weights on a psychopathology factor. If you use PLS for feature extraction, then you just need to be careful to use it in cross-validation. And so the way you would do it is you first separate your data into a training and a testing set, so both your imaging and your behavioral data. Then you compute your PLS on the training set. You decompose your correspondence matrix, and you can compute the latent variables for your training set. And then you take your test set and you project them on the saliences of your training set that we got here to get latent variables from your testing data. And these latent variables can then be used as features for classification. So this is the end of my talk. Here are a few references if you want to read more about PLS, or also some related methods. So the first paper here by Krishnan is a review that's very comprehensive and comprehensible at the same time. The second paper is by McIntosh, and it's the paper where he explains how to apply PLS to neuroimaging data, and a lot of the steps that I've explained before come from this paper, come from him. And in these two papers, we have descriptions of PLS and also other methods that are very similar, such as canonical correlation analysis, as Jonas was saying before, or also ICA methods. And finally, we have an in-house MATLAB code, which I'll be demonstrating on Wednesday at the Open Science Room, which will be available very soon on this website. And I also want to mention the Rotman-Baker's MATLAB toolbox, which is also openly available and which is quite user-friendly, and that can also take many imaging inputs such as task fMRI, structural MRI, or PET data. Oops.